Alonso, ¿me escuchas?
Sí, aquí soy.
Listo, Alonso.
No, es que lo que pasa es que tiene que ser en el computador. O sea, se pierde la conexión con los otros. Sí, o sea, que si ya lo tiene en PowerPoint, se la USB y se pone allá.
You can use PowerPoint.
Sí, sí, apunte. No, English, no.
Oh. Pleasure to be in. I think you can hear me without this, right? Can you hear me? Okay. It's a pleasure to be in Cali, Colombia, and I'd like to thank Professor Torres and Professor Maria Gomez, Maria Elena Gomez de Prioto for having me here. And I'd like to talk to you about superparamagnetic nanoparticles in biology and medicine. But I think I'm essentially
For the streaming, you can please use the microphone.
Okay. And so I'm going to give you an overview of a little bit generalities first to help you understand the essential aspect of magnetism, because we're going to be using essentially superparamagnetic nanoparticles. And in a way, this will help you understand the, the talk. And if you have any questions, please do not hesitate to interrupt me and ask me the questions. Don't wait till the end. Okay? So, well, one of the main objectives of our work is to use nanoparticles. And you know, when nanoparticles started, they were all taxed as being very toxic. And there were many, many studies about the toxicity of nanoparticles. And several years after the appearance of these nanoparticles, well, they got kind of like a clean bill of health. But so it's in a way ironical to use nanoparticles to fight some diseases, and especially cancer. So why cancer? Well, this thing appeared recently, and as you can see down here, it's adopted by Horizon 2020, which is the EU framework program for research and innovation. It is also highlighted by the National Science Foundation in the United States and by other societies, of course, but in a way where the money comes is what becomes very important in funding research. So anyway, since the two major places which sponsor research, such as the NSF and the EU framework, start putting money into these projects, so the research will be blossoming in it. So what are the grand challenges of the society in the 2014? Well, as you can see, health and well-being is number one. And then we have the clean environment, and then the secure, clean, and efficient energy, including smart transport. Then, of course, sustainable agriculture and water research. And as you know, water is disappearing in some continents on Earth. So there is an urge and necessity to economize on water. And then innovative and secure society, we're all aiming into developing such a society. Now, given that, well, among all of these things, you can see that cancer is a big public health concern. And here you have the numbers, which were taken by one of my colleagues, Professor Eloisa Bordello. And she kind of gave the same statistics, which is quite impressive. So 0.5% of the total population in Europe, which is about 3 million, let's say, and a half new cases have been found in 2012 relating to cancer. The European population increase is about 1.1% per year. So it's quite a big increase of new cases. And then if you go back to the United States, well, it's surprising, but it's about the same statistics. So you have here 0.5% of the total population. And the increase of the American population is higher than in Europe, which is not surprising, right? And so it's about 1.3%. And these facts come from the American Cancer Society. So you see the number of new cases expected every year is quite tremendous. So these issues about cancer must be addressed in a way, and the hope is to find better solutions. Well, as you know, most common cancer treatment is, well, you can classify them in two categories. One is radiotherapy, where you calculate a specific dose of radiation, and you deposit it into this tumor tissue. And then, as you may have heard, or as you know, this is very good for localized lesions. The other very common treatment is chemotherapy, where essentially, I could use that, it's chemotherapy, where essentially the patient is subjected to a cocktail of chemical treatments, which in a way may help in saving some specific problems, but then there are all the side effects, which sometimes are quite nasty. So where do we come with, as I said before, well, it's kind of ironical to use nanoparticles to help in biomedicine. So what do we mean by nanoparticles? Well, these are particles which have a controllable size. So it goes from 1 to 10 nanometer. And for example, the size of these nanoparticles are comparable to the size of cells, viruses. You know, it's not way off. And well, and proteins. So in a way, you can get close to the biological entity of interest with these nanoparticles. And then, well, the idea is to coat them with biological molecules so you can bind them to another biological molecule. And, well, how do you handle it? So the concept was to use an external magnetic field gradient. So in a way, the dream is to be able to take this drug, what we call a package, which is an anti-cancer drug or radionuclide atoms, and then you go to a specific part of the human body, so you don't in a way damage the entire body. So you just take it, you deliver it. It's like a taxi delivery. You go from A to B, and you don't go anywhere else. And then, well, what we would like to do also is to be able to heat it, because some of these treatments that I'll show you later show that when you're heating it and with a time varying magnetic field, you can then use these entities as a hyperthermia agent, and you can deliver, again, the toxic amount of thermal energy to the targeted bodies, such as tumors, or alternatively, you can use them in chemotherapy and radiotherapy. And this is where you could heat the tissues and then be able to address the questions of cell destructions. And finally, we would like that these magnetic nanoparticles, we would like to use them as a magnetic contrast agent, for example, in techniques such as magnetic resonance imaging. And this could help in getting a better picture about the whole system. So what are the biological applications of these magnetic nanoparticles? So I grouped here some of the most recent applications. And as you can see, we would like to be able to use them in hyperthermia, which I went through. So you have heating to have, and then in drug delivery and vectorization. And actually, this is what we call this taxi delivery. You take it from one place to another place without affecting the rest. And you can do magnetoffection, magnetic immunoassay, and then you can also use them as a contrast agent in the MRI. And here we have grouped some kind of papers where they have been discussed, and I would like to draw attention to the PhD thesis of one of my students, Irena Milosevic, where essentially this was the beginning of the use of such nanoparticles. Well, I decided to go through some explanations, as I mentioned before. What do we mean by for example, ferromagnetic or superparamagnetic behavior, for those of you who aren't physicists or in material sciences, well, it's all a question, again, of size. So large particles of the order of micrometer, and you have this in the figure on your left hand side, well, these large particles of the order of micrometer, they have a multi-domain ground state, and therefore they have a very narrow hysteresis loop. And then the smaller particles, smaller particles have a single domain ground state, and so they have a broad hysteresis loop, and these are the ones which are on the outside. So these are the ferromagnetic. Now, if we move to the curve on your right-hand side, this corresponds to a superparamagnetic nanoparticles, and in this case, they are smaller sizes, usually less than 10 nanometer, and the magnetic moment of the particle as a whole is free to fluctuate in response to the magnetic field applied. And as you can see here, there is absolutely no hysteresis, and it's a simple and it's clear, and there is a zero coercivity. Now, both the ferromagnetic and the superparamagnetic particles have susceptibilities which are higher. And I would like to point out that to you, that they are significantly higher than normal paramagnets. And so all the individual atoms are going to line up, and their susceptibility is much higher than the susceptibility, for example, of blood and other biological materials. And this is an important advantage that one can use. And by the way, I like to always point out that I have taken it from a very famous paper by Quentin Pankhurst from London, England. And anyway, I like to point out that this is a very small journal, JPhysD Applied Physics. It has a very little impact factor, but I believe that it's one of the most read papers. So, you know, it's not always very important to look at impact factors. And so now we move on to talk a little bit about the MRI contrast enhancement with magnetic nanoparticles. And here, again, I have given the whole physics behind it, but I think what I'd like to point out is that what is very important is that when the superparamagnetic nanoparticles are associated with the target cells, they become normally saturated, magnetically saturated in the normal range of the applied field used. And in a way, what happened, we can have a substantial locally perturbed dipolar field, which leads to a marked increase in the relaxation rate 1/T2 square star. And if you look there to this equation, you see that 1/T2 star is larger than 1/T2. So it even makes it larger. And this is something that we could use again in the applications. Now, as we said about this imaging using magnetic nanoparticles, for example, in this case, what we're talking about is having, again, these superparamagnetic iron oxide nanoparticles, which have a hydrodynamic diameter over 50 nanometer, and it can go to 150 nanometer. But these have been developed as a contrast agents. So what do you do with them? So when you inject them in the liver, for example, or in the spleen, there is lots of the plasma life is lower than 10 minutes. And so what happens is it can give you a very good contrast. And I don't know here if the quality of these figures is visible to you, but I could see it, you know, on my screen. As you can notice on the left-hand figure, what you have is you see spots like you know, these two, and then you see something here, and it's all. But then when we use the magnetic nanoparticle, it becomes very clear where is the affected metastasis, and these are these two regions, and the others are okay. So anyway, when we're using these spions in the magnetic resonance imaging, the images that you get are much more revealing of the details, and they give you better information. Okay, so now we talk also about the forces exerted on these magnetic nanoparticles. And in a magnetic field gradient, a magnetic particle, well, you have this equation for the force. And as you can see here, the force is proportional to the magnetic susceptibility, which is given by psi. And this force is really the basis for the biomedical applications and drug delivery and in magnetic separation. So anyway, what happens in a typical magnetic drug delivery system, what we like to think of is that if you put a magnet outside the body, and this magnet, it creates a magnetic field gradient, which will capture the magnetic carriers flowing in the circulatory system. So anyway, this is what's being explained in this diagram on your left-hand side. And as you can see, the magnetic nanoparticles with the blood vessels, you know, and the whole thing, thanks to these forces, they can get lots of displacement and reach exactly the areas we want. And there is nothing like a figure to illustrate what one is talking about. You can see these are more recent figures, and the contrast is actually quite impressive. So here we have a magnetic targeting of the drug delivery. You have the magnet, you have the tumor, and then you have your magnetic nanoparticles with the drug, and this is conjugated inside a ferrofluid, and you can see what you can target it exactly to the tumor. And this is the magnetic drug delivery. As you can see, the 2 feet are still okay to some extent. And this is when you use the conventional treatment, you get all the extra side effects. Now, the other thing interesting is what I was talking about, the magnetic resonance imaging apparatus. This is, again, this contrast that we first didn't see very well because the picture was black and white, and it was kind of like the first demonstration of these effects. So historically, you know, they were the first to come up with it. And in this work of 2007, you can see the contrast before the administration of the magnetic nanoparticles. You couldn't distinguish at all which is the part which is not healthy and the others. And this is after administration of the drug with the magnetic nanoparticles. Anyway, you could tell where is the tumor or the metastasis. So I hope that this is convincing you a little bit. And finally, the other thing that I'd like to mention very quickly, because it's related to work happening in Copenhagen in the group that I mentioned before. And this is where, as an alternative to the drug delivery, what one would deliver is a gene delivery. And this is a little bit more sophisticated. And actually, it's this figure is taken from the work of the team of Professor Dobson And as you can see here, what's happening is you have an oscillating nanomagnetic transfection. So here what you have is a plasmid DNA or RNA, and then you attach them to these magnetic nanoparticles, and you incubate it with cells in culture. And thanks to the magnetic field and small oscillation, what you end up doing is transferring a gene delivery instead of the drugs. And finally, as I mentioned before, the idea of heating these magnetic nanoparticles is something which is very important. And here I give you a little bit of the physics behind the heating of these magnetic nanoparticles. And in a way, when a superparamagnetic nanoparticles is subjected to a magnetic field, while they experience a heat generation P per unit volume, which is given by this equation. And in a way, what you have here is the frequencies which could change. And in a way, what we have to always remember is that the range of frequencies and amplitude which are tolerable by a human body is the frequency should be about 0.05 to 1.2 megahertz. And then you have the field should be about 0 to 15 kiloamp per meter. And anyway, this is the basis for biomedical application of the hyperthermia and the enhancement agents during chemotherapy and radiotherapy. And then, you know, we can also talk about the rotation of these superparamagnetic particles. And
how many grades are temperature change?
Well, it has to be, you have to always remember that the temperature change, thank you for the question, the temperature change cannot be extremely high because the human body cannot support such high temperatures. So usually what they aim for is a maximum of 45 degrees centigrade. This is what, you know, kind of like the aim. And so if we can go back to this heating, the second part of it is, as I mentioned before, is you have to remember the relaxation. Actually, I think it's written somewhere here. No, it's not. Yes, is you have to, here what we show is the calculations, the theoretical estimation, and then the experimental data. And as you can see, you go first through the Néel relaxation, and then as the particle diameter increases, well, you go through the Brownian relaxation. And what we call the SAR, well, is going, the SAR is, sorry, just a specific, sorry, I'm completely, a specific absorption rate. Is going to depend on both the field H and the frequency F. And in a way to compare the results with different groups, from different groups, what we do is we use a parameter called intrinsic loss parameter, and it's defined by this equation. So now if we move to the, and this is in a way the illustration of this magnetic targeting and the gene delivery. And I think this is a picture taken from a center in Berlin where they were the first to do it. And in a way, it kind of gives you a graphical picture of how the things is applied. Okay, and now when you think about this movement, well, there is two ways that one can apply these magnetic and nanoparticles and then ensure a delivery. One can do it in a very gentle magnetic heating, So you do it gently. So what happens is you go through a volume shrinkage, a slow drug release, and the process is reversible. So you do it in a gentle way and one wait for it. And the second alternative is to use an intense magnetic heating. So you apply everything kind of like, you know, one time, and then let's see what happens. And in this way, you cannot go back and repeat the process. So you do it in one shot, and then everything, you deliver it, everything at the same time. And it is called irreversible procedure because you cannot go back. And this leads to a volume shrinkage under the effect of something so quick. And you get a structure disruption, and you have an instant, a huge drug release. So all this drug release is released at once. And anyway, this is something which is commonly used. There is really no strict rules about it. It's kind of on a trial basis. And well, if we continue on this, anyway, here, what we're having is a magnetic gel bead. As you can see, we have the magnetic nanoparticles. We have the permanent magnet, and what we do is, under the effect of these vibrations, everything gets released, and depending on the speed, what you have here is the plots of the release per milligram, in this case, of this a drug called ketoprofen. And as you can see here, without any rotation, you have a straight, but you don't go far in 20 minutes. And then if you have a very strong rotation, well, you get to the maximum in few minutes, and the others responds very, you know, and in between the two. So the quantity diffused as a function of speed, in a way, this shows that one can have a handle on how to release it. And in a way, this is affects the hyperthermia therapy. And in this case, it's again, you know, you do heat the magnetic nanoparticles, and again, you maintain it between 40 and 43 centigrade. And in a way, this hyperthermia therapy is used a lot in treating cancer, because it's not as, let's say, doesn't have as bad reactions and bad consequence follow-ups. And so in a way, if one can combine the use of radiotherapy and hyperthermia, this leads to a better chance of getting a success rate, higher success rate. And these first clinical trials actually were again done in this charity hospital in Berlin. And this is, you know, in 2007. So, okay, so now what I would like to pass on after this introduction about a little bit combining let's say, physics to help understand all these procedures in treating cancer via drugs and having magnetic nanoparticles. I'd like to talk to you about how did we go about it. And so in this thing, it's we have to have a multidisciplinary research. So our team was composed of physicists, chemists, and biologists, and pharmaceutics. And this was possible because we had a hospital where there is research going on, so we could use their pharmaceutics. So we were kind of like multiple disciplinary, and I think it's the best. So some people did the cooking, which is the synthesis, the purification, and the formulation. And essentially what I'd like to present to you is what we did is we had to go and synthesize these nanoparticles And then we had to use a polymer. And there is a French expression which you say you dress these naked nanoparticles. So you use a polymer to cover them. And as you may know, polymers look like spaghetti, right? So you need many layers of this polymer to cover these nanoparticles. And then, of course, we had to use a drug of some kind, any biomolecule. And the idea is to put this drug and bury it inside. So, you know, when you're going to put it in a body, the body doesn't touch directly the drug, but it's being separated from it by layers of other things like the polymer, which is biocompatible. And then, of course, when you make something like this, you need to do some kind of characterization. And as I have seen this morning, you have wonderful equipment for characterizations in the department. And we were lucky also to have some available. So I'll show you the results of our physical chemical characterization. And then you have to go this step further where you have to do biological evaluation and imaging, as I talked before. So, well, okay, so what we did is we focused on iron oxide. And as you may know, iron oxide is quite a complicated material because the iron has several valences. Then you have several forms of the iron oxide. But what we tried to do is focus on the properties of paramagnetism. And then we aimed at having superparamagnetic iron oxide nanoparticles. And it's a shame here you don't see this. Actually, what's present here is a black liquid. And then you put a small magnet. And by moving the magnet, the liquid moves up with it, which is kind of nice, right? So we always like to show it. Okay, and then so we stayed at small size. We stayed less than 10 nanometer with a hydrodynamic diameter of about 60 nanometer. And we aimed at the paramagnetic. And then these are the biological molecules that we considered. And we did the coating actually in two ways. We did some with polymers, but I will not talk to you about, we also used metallic like nano gold, nano silver, because these are kind of not toxic and they have interesting properties. And the other thing we used, of course, the fluorochrome and the drugs. So essentially, this is kind of like a nice picture because it shows you everything which went around these nanoparticle cores. And it's amazing how nowadays one can achieve such a construction experimentally in the lab. So this is the method we used. We went from, we used iron 2 and iron 3 salts in an alkaline medium. And well, both of them co-precipitate and you get the nanoparticles. And then we put them, they come into a colloidal suspension, essentially. And the aim is to keep them well separated and not have them gather together. But I think this is now very well controlled. And we determined the hydrodynamic radius by dynamic light scattering. And so we also used TEM and DLS, and we got all these numbers. And well, this is another interesting thing which could be of interest to some of you who are working on iron oxide, is that when you, we looked at the composition at the, sorry, the slides, some of them are in French, but they are easily readable, right? It's chemical composition. And we use FTIR spectroscopy. Why did we use that? Because anyway, when you start with this iron oxide and you make it into nanoparticles, we don't know what's the valence of the iron. So anyway, with this FTIR, you can see here that there are these two extra, I don't know if I can call them peaks, but they are more like minima. And this is a signature of the magnetite and the maghemite. And then we did also some actually spectroscopy, Raman spectroscopy. And by fitting, we believe that this is the peaks corresponding to the maghemite and the magnetite. And then by fitting these, we were able to tell how much magnetite and how much maghemite we have. And I would like to, as you can see, it's essentially magnetite with about 35% of maghemite. Now, this is a very interesting concept because, as you know, it's you cannot tell the difference by X-rays, okay? Because the X-ray spectra of both iron oxide, a maghemite or magnetite, well, it's essentially 100% identical. So from the X-rays, you cannot tell. And there is another method that you can use to tell the difference between the maghemite and the magnetite is Mossbauer spectroscopy. Of course, Mossbauer, you know, you can tell the state of iron, but you have to have a Mossbauer to do this. So anyway, using something standard that everyone has in a you know, available, like a Raman spectroscopy or an FTIR spectroscopy, will help in telling you exactly how much of each you have. And tomorrow I'll get back to this point because it's quite interesting. There is also, since the X-rays, you cannot say X-ray in general, but it's the X-ray, you know, as we practice it, like diffraction X-rays would not give you any difference. But if you go to a synchrotron, and I don't know if you have one in, I think there is one in Argentina, Brazil maybe, or Argentina, right? There, anyway, there is a technique which takes like 1 second, and it's called exanes. And this will give you in a very, you know, unambivalent way the amount of maghemite and magnetite. Anyway, so now what we constructed here were just simple nanoparticles. I'd like to show you something we just published recently. And here what we did is we used nanorods, because there are some things and some procedures which become much more efficient if one changes the shape of the nanoparticles. So in this case, we used nanorods, and we're looking, as you can see here, there is the X-ray, the DRX, and then you have the TM pictures. And this is another alternative to spherical nanoparticles. Okay, so now if we go back to our cooking, so what do? We have here the spions, and then we have to dress them up. So what we start doing is we put, first of all, the PEG, which is the polymer, only 6,000, and this PEG is very highly biocompatible. And we add to it something called folic acid. And as you can see, this folic acid is represented in this orange color. And I'll get back to why do we use folic acid. And then we also used some PEG 5000. And this is a neutral polymer. And in a way, it helps in maintaining the colloidal stability of these nanoparticles so they don't grow by touching each other and getting together, and also to help the stillness. And the drug we used is a doxorubicin, so we had to hide it inside. And this is a cancer chemotherapy, and it helps in the fluorescent monitoring. And this kind of gives you an idea of the synthesis which was carried. And then we came up with our samples, or our designed the drug very well, camouflaged and so on. But this is, we need again to go and investigate if this is the case. So I'd like to, in this case, what we wanted to do is look at the surface and make sure that none of these drugs came to the surface, and what's on the surface is what we believe is. So we used actually two techniques. One is the XPS, and I think you have it here. And the XPS allows you to look at surfaces. It's also element specific, and it has, you can in a way tell numerically how much you have on the surface. But the problem with it, it's element specific, but also it's only can see the surface up to 10 nanometer from the surface, and you can squeeze it into 2 nanometer. But then we used another technique, which is a very expensive instrument and very delicate to handle, and it's a time of flight SIMS, and essentially it is like an XPS, but it does the ultimum surface. So you can probe at just 0.5 nanometer below the surface, and you can go to 2 nanometer if you wish. But the problem with it is that it allows to be molecular specific, but it is essentially a qualitative evaluation of the surface. So you see what's on the surface, but it's very, very difficult. It's possible, but takes lots of time to come up with precise numbers. But all what we wanted to check in our case is to make sure that that the drug didn't make it to the surface, and so we could look for the elements of the drug. So this is the spectrum of spion with DOX. Again, DOX is the drug, and the PEG. And we looked by the XPS spectrum, and at 10 nanometer, what we only see is we see all the elements. We see carbon, oxygen, nitrogen, and silicon. And in a way, we can also calculate the percentage of nitrogen in the spion, DOX, PEG, and in the other one with the folic acid. And at 2 nanometer, we don't see any signal of the nitrogen. So, and the nitrogen comes actually from the drug. And when we go to the spion with the DOX and the PEG and the folic acid, well, we have results which are, again, from the XPS at a distance 10 nanometer from the outer surface. We find the same elements, and we have the same percentage of nitrogen. And at when we go to only 2 nanometer from the surface, we have the percentage of nitrogen which increases. And you know, this shows all the calculations related to the treatment of the data. And this nitrogen increase because we are seeing the folic acid. Now, when we go to the TOF SIMS, this is the kind of spectra that you get, and you have to analyze it. And what is interesting is we looked at the folic acid by itself, and that we took from the literature, and this is what the results you get. And here we have the PEG 5000 and so on. So anyway, we could identify all these peaks, but again, we didn't go into the calculations. And then from it, what we came up with is that the outer surface in the case of this association, well, in the case where we have the PEG, well, we only see the PEG, And when we have the folic acid, we see the nitrogen related to the folic acid. So anyway, these measurements convinced us that we had exactly what we aim to have. And basically, we have this nanoparticles, and the DOX is inside. And then all what is outside is the PEG and the folic acid. So there is no danger of having the DOX being directly in contact with the tissue. Now, the other thing we use to make sure is we use surface-enhanced Raman spectroscopy. And the advantage of this technique are listed here. So you can identify molecular fingerprints. It's not invasive. It's non-destructive. It's really a fabulous modification or, say, sensitization of the Raman spectroscopy, but it's related to what's happened to the surface. And in a way, this helped us identify that the results of the XPS and the TOF-SIMS were correct. Now, you may want to ask me, why do we use two different techniques? Well, it's very simple. What I forgot to tell you is in the case of all these surface things, like the XPS and the TOF-SIMS, we had to take our solution, which is liquid, and turn it into a powder. So there is always this worry that when you go from a solution which is liquid and you make it into a powder, that you may have changed how things were wrapped inside. And with the surface-enhanced Raman spectroscopy, we could look at it as it is. But of course, we cannot get so close to the surface as with the TOF-SIMS. And as I mentioned here, this technique is ideal for in vitro and in vivo biological samples, and it has a better sensitivity than fluorescence. Okay, so this is the SIRS spectra, and what we looked at is we looked at doxorubicin. Again, this is the drug, at the folic acid, and our combination of spion, DOX, and PEG, and folic acid. And what is the most important for us is that you can look at it, there is absolutely nothing in the spectra which is taken from the spectrum of the doxorubicin. So in a way, this again confirms in the liquid sample that we do not have doxorubicin on the outside. So now with these results, we looked also at the colloidal stability. And well, as you can see here, everything is in order. We have absolutely nothing which is abnormal. And you can see with the folic acid and without the folic acid, there is barely, there is some changes, but nothing in what concerns us. Okay, and so now these are the results of some testing we did. And well, this is here we looked at the activation test as a function of the nanoparticle surface area. And it's here, this is with the spions alone. And then the red is the spion with the DOX and the polymer. And then in blue is the spion with the DOX, with the PEG and the folic acid. And as you can see, the folic acid, if anything, it doesn't perturb and it doesn't, add any significant, but anyway, it's handy to have it. And anyway, it is, we have the same activation test. But now when we look at the macrophage uptake, I'd like you to just look at the red and white as a function, red and blue as a function of time. So as you can see, for an incubation time of 60 minutes, what we have is when we have the folic acid, it's little. Then if we go to 120 minutes, well, we have the same fluorescence, we have the same macrophage uptake, and you can see here they are both equal. And if we wait much longer, 240 minutes, well, you can see that the folic acid had some influence and improvement over the spion dogs with PEG without the folic acid. And this was the, we looked here at the tumor cell uptake, and this is again, you know, the diagram of what we have synthesized. So we have our 10 nanometer dogs, iron oxide, the spions, and we have the dogs around it. And then we have the polymer and the folic acid. And this is the cell membrane. So these are the pictures taken by confocal microscopy. And anyway, it shows you with the spions and the folic acid and without. And we see a much better results with the folic acid. And when we look here at the percentage of spion and PEG as a function of time, again, the presence of the folic acid brings a positive element to the pictures. Now, what, anyway, I'd like to bring also to you some very recent work, which is still unpublished, but Eloisa Bordalo, she said I could mention it. So anyway, this is an alternative approach. And again, what here is a bio-nanocomposite, which is used in the cancer treatment. And as you can see here, what they have done is the magnetic nanoparticles are manganese and zinc ferrite, and they are well, shown here. And what they do is similar to what we're doing. They have a polymeric coating around it. And the drug in this case is this paclitaxel. And it's also incorporated inside. And what they use is some nanocrystals made of hydroxyapatite. And with this, they are targeting, anyway, the cancer cells. So what we learned so far, well, the main aim was to design an easy and economical protocol to for the preparation of, in our case, of the iron oxide nanoparticles. We performed a complete and thorough characterization of these nanoparticles. And in a way, one needs to have every possible way of characterizing it and of testing everything. And we used analytical methods by combining them to try to look at the chemical structure and properties of these nanoparticles. And well, our preliminary biological studies showed that the functionalization with the folic acid increased their uptake by MCF7 cancer cells. By the way, these MCF7 cancer cells are breast cancer cells without altering their properties. Well, I think at this stage I have talked enough. I'll stop and I'll have to ask you for some questions to make sure that I was able to deliver the rest. Thank you for your attention. more is coming tomorrow.
So far, this type of research is in the basic stage. How far is the use of these methods in a clinical testing? And how far is the approval of the FDA for the use of nanoparticle? And the second question I have is, how, if you use nanoparticle in a normal person, not in a dog, something like that you show, what happened with the particle after this treatment? How toxic is the particle for the other tissue or other organ in the human?
Yes.
Okay.
Yes. No, anyway, what we avoided doing, we started first, as I mentioned, with silver and gold. And it seems like silver is okay, but there is some minimum amount of toxicity in it, so we dropped it. But we had very nice papers, and I think we understood a little bit more about nanoparticles of silver. And I think gold is also in the same rank, so we dropped it. Now, iron oxide, I think, is not at all toxic. So this is in the normal state. Now, when it becomes nano, sometimes it changes. But here, we're not talking about nano, we're still like, we're not in angstrom, we're, like 10 nanometer or something. And then, well, every chemical has problems, but the question is to level it with the alternative, right? And I think iron oxide is not as bad as other things. So iron oxide is accepted, let's say, between, even in this nano form, if you go and look at, the how is it called? The things that FDA has in the United States. So it's something acceptable. The second question is, did we perform enough tests? Well, it's very, very complicated in France because there is a society who protects the animals. So now and then, every time that you have a lab which has mice, and I always wondered why use mice and not rats. But anyway, they use mice, right? So any lab which has mice, there is sometimes demonstration outside and so on. So it's a little bit difficult. But then there are other places where you can do these kind of trials. And we worked, as I mentioned, with hospital in Tours, which has research and so on. So we were able to do some of these testing, but honestly, it's not something that we can continue. And the idea is that it will be picked up by other teams which are more qualified than us in it. But, anyway, it opens the way to alternative medicine, alternative ways of dealing with cancer and how to approach things without destroying other parts and so on. So on the scale of things, it's not as dangerous as others. Thank you.
What about the magnetic the hyperthermia? Magnetic hyperthermia is
magnetic hyperthermia.
Hyperthermia.
Yeah. Well, I think it is, and it is being used because this magnetic hyperthermia that I showed you, it's in a clinic in Berlin. So I think it's being, it's used, and it's kind of, again, it's not very offensive and destructive as much as other techniques. Yes. So this is the answer. Well, I spent time in sorry. Well, Well, actually, this is something we would like to pursue, definitely. And, you know, we're looking into it. And my collaborators in Tour are looking into it extensively. And I was going to say that I'm an invited chair professor in Shanghai, in a place called Suzhou, which is half an hour from Shanghai. And they do, I go there, and actually they are working on similar approaches for drug delivery of cancer. And there they have the possibility, you know, they have many, many mice. And their rules are different. I mean, mice in labs, in the lab. Sorry. Thank you for smiling. Yeah, so, you know, they have larger quantities of animals that you could try these things on. And they are working on similar approaches, and it's quite positive. And the other thing is there, when you give a seminar, the students are not allowed to leave without asking questions. So maybe we should impose it here. You're not a student, but thank you. So
the nanoparticles, iron oxides. Do you have any pictures about the porosity of those nanoparticles, the morphology?
morphology. I think, yes, we did them because there was a PhD thesis, and I'll be happy to show it to you.
Okay, thank you.
Yeah, we did. And actually, what is really also interesting, and in the case of these, you know, the iron oxide is heavily used by other people also.
Yes.
And what they are trying to do is to confine it. You know, instead of going through all of these polymers and, you know, this heavy synthesis, as they are trying to just load it in some nanotubes or put it maybe in zeolite or something like that, where you could release it and look at it. But then the question is how much the confinement is going to affect the properties.
And you have checked the difference between nanorods and nanoparticles?
Yeah, this is a very good question. We haven't yet done it, but this is something we really want to do, because these nanorods are interesting also. In this case, we did them only with magamite. No, with the, what is the form? Is the well, it's with another form of iron oxide. And actually, we're very interested in looking into it with more details, because this form is kind of like a geologically or available, so it's kind of 100% natural. And we haven't yet looked into it. But this is, again, it's a possibility of confining it without as much problems as with the spherical one.
Okay.
Thank you. Oh, sorry.
Okay, so I guess that in the moment that you synthesize the nanoparticles, the size of the particles is not equal. So how can you control the size of the nanoparticles? Because some of them could be more, could be bigger than the other ones. So
Right.
how
Right.
could you do that?
Right. Actually, this is, you know, this early synthesis were done something like 3 or 4 years ago, where we didn't have as much control over the sizes, and now it is, we're aiming at having a much more controllable size. But there is a limit to how much you can do it, because, you know, in a way, what you have, I think also one of the things is when you're going to get them in the human system, there is some kind of pressure which is going to be exerted on it. So we would like to look at the pressure, and one of the effect of pressure is maybe is to get them to increase in size. If you get two of them together, you know, despite all the precaution that we'll take for the colloidal stability. So this is one of the questions that we want to do, and improve the monodispersity of these nanoparticles. Yes.
So are you able to use some kind of separation method, or I mean but by filters or something like that, that could separate the nanoparticles by sizes, or nothing?
Actually, until we have really a definite answer about the effect of this which is still quite small, the polydispersity that we have, until we have, you know, a proof that it matters, it doesn't pay to go into extremes to, you know, to do such steps, because, you know, it's what we want is to have them remain superparamagnetic, because we're taking advantage of this, and, you know, the frontier between both the superparamagnetic and the regular paramagnets, not superparamagnetic and the ferromagnetic, is not so, you know, it's quite large. So by having it coincide is not going to change the magnetic properties drastically. So anyway, yeah, it would be nice to do a perfection and having a very good monodispersity, but I don't think we'll gain a lot in return. But I think playing with these valences is always an interesting thing, you know, changing the quantity of the Fe2 to the Fe3 plus could maybe have some alternatives.
Hi, in RMI resonance magnetics imaging applications and tumor cancer cells in the brain, application for neuroscience, neurophysics. What is the perspectives, the futures, Anderson? Anderson?
Yes.
Bueno, the following, de marcaje, el seguimiento de las nanopartículas, si me puedes ayudar con eso. ¿Sí? ¿Cómo haría el marcaje? El marcaje para poder seguir las nanopartículas en tumores cerebrales.
Yes, actually, I'm happy you asked that, because what I'll be talking tomorrow is reviewing work of my colleagues at Argonne National Lab, who looked specifically at the brain cancer, but then they used a different approach. What they used is microdisc, and I'll be talking about it. But we somehow focused on the breast cancer, because maybe it was the easiest one to find colleagues in Tours, you know, which is close to Orléans, and which have expertise in this kind of cancer. So this is why we focused on the breast cancer, because, you know, every cancer, especially when it comes to the brain, is very different and very, you know, quite, you know, you need to have colleagues in this field and so on. We haven't looked at the brain cancer. Thanks. Yeah.
Okay, thanks.
Had you used some antibody or label the nanoparticles? In that way, you can use a target for the nanoparticle.
Yes, I think this was done by the people who did the testing. I think they label it, but I really don't know how because I didn't follow this in details.
Okay, thank you.
But they do labeling. I think this is part of it. Yes.
After the nanoparticles accomplish their functions, have you followed it? What happened with those nanoparticles after they accomplish their function in the body?
Actually, this was one of the questions which we always asked, and in a way, you know, we didn't follow it, but the logic of it is that it will be eliminated like anything which goes in the body. You know, what goes in goes out, and so we assume that if this is to be applied on a normal patient and so on, you know, it's going to be eliminated like anything which goes inside the body. And in a way, it's not, you see, our biggest worry was how to camouflage the drug. So it's not sitting on the surface, kind of like, you know, when you deliver it here and you imagine you guide it to the place where you have the cancer. So in this route, it shouldn't interfere or interact with other things. But, you know, we just assumed what goes in goes out. And again, iron oxide is not at all, you know, it's not toxic, you know, it's FDA approved, even at the nano scale. So.
Okay. ¿Alguna otra inquietud? Pregunta. Contar lo que hacemos. Ok, entonces pido un aplauso para la profesora. That tomorrow at the at 10 hours. De la
We're saying tomorrow some people have classes in 2 to 4. Just asking if there's video or way to follow you tomorrow. They can make it because they have class tomorrow. I think that they will share the video.